Good morning to all. Today topic is cholesterol biosynthesis. I am Dr. Lenin, Department of Biochemistry, Assistant Professor. And see that one by one, this topic I have to tell about for this. What are the important specific objectives from this topic? And it, first, you have to discuss about for the structure of the cholesterol, and second one is the metabolism and biosynthesis, and third, what are the function and sources of cholesterol, and utilization and degradation and regulation. So these are all the most important and specific learning objectives from this topic. How to write for the structure, how to draw the structure, how to metabolize it for the cholesterol, and function and sources for the cholesterol. And see that one by one and this topic. So this is the introduction about for the cholesterol biosynthesis. Uh, you know that uh, you know that and cholesterol, it is a major key role in the animal tissues. The cholesterol is presented in tissue and in plasma, either as a free cholesterol or as a storage form combined with a long chain fatty acid that is called as for cholesterol ester. In the plasma, both forms are transported in the lipoprotein and by removed from the tissues by the plasma, it is lipoprotein and transferred to the liver, and it is are eliminated from the body either the unchanged or after the conversion to the bile acid in the process known as reverse cholesterol transporters. Again, this is the important introduction about for the cholesterol. The cholesterol is the major steroid in the animal tissue. It is strong. It is for presented in the tissues and the plasma. And most important thing is both the form are transported in the lipoprotein and remove the tissues by the plasma high density lipoprotein and transferred to the liver. Its varieties are eliminated by this from the body either unchanged or after the conversion to the bile acid in the process known as reverse cholesterol transport. This is the cholesterol structure. It having for this four non-aromatic rings and named as A, B, C, D, and 27 carbon compound are presented, and one double bond between for that C5 and 6, and the cytine having presented for this, and one more hydroxyl group at position for the carbon 3. So that having for the 27 carbon compound, and it is a one double bond are presented for this carbon 5 and 6, and one hydroxy groups are presented for the carbon 3. So cholesterol, it is derived from this diet and de novo synthesis, and from the hydrolysis of cholesterol ester. It approximately for the liver and the intestine account for approximately for 10 percentages of each of the total synthesis in the human, and which should all the tissues it containing for nucleated cell or the cable of cholesterol synthesis, and which occur in the endoplasmic reticulum and the cytosol. And this is the slide I show that, and the cholesterol maximus are derived from diet and the synthesis of de novo and hydrolysis for cholesterol ester. And one more thing, 10% of each total synthesis in the human body, and usually for all the tissues containing for nucleated cell is are capable for cholesterol synthesis and which occur in the endoplasmic reticulum, the cytosol. And cholesterol is most occurrent steroid in the human. It performs a number of essential functions. So it have the cholesterol. Most of the functions are presented, and see that one by one is it is a major constituent of the plasma membrane, and it is and of the plasma lipoprotein. It is a precursor for the bile as salt, and it is also precursor for the steroid hormone that included for adrenocortical hormones and six hormones and the placental hormone and extra. It is also the precursor for vitamin D. It is a precursor for the nerve transmission. So that is all the important function for the cholesterol. First one is it is a major constituent of the plasma membrane, and it is a precursor for bile salt, It is also precursor for the steroid hormone, like for the adrenocortical hormones, sex hormones, placental hormones, and extra. It is also the precursor for vitamin D. It is a record for the nerve transmissions. The steroid hormone, it is all the steroid hormones are derived from cholesterol. It is the cortex of adrenal glands. There are two classes of hormones are synthesized. One is for mineralocorticoid and glucocorticoids. In the male and female gonads, for example, for the sex hormones are producers. One is for this progesterone, androgen, and the estrogen. These are all are it is synthesis and a record to precursor. These are all the hormone. It is a synthesized and it also help the precursor of cholesterol. This is for the cholesterol biosynthesis in adolescents. Slightly less than half of the cholesterol in the body you can do or record for the derive from the biosynthesis of de novo. The biosynthesis in the liver account for approximately for 10 percentages, and in the intestine approximately 15% it is synthesized from the cholesterol of the amount of product each other produced for the each day. The cholesterol biosynthesis pathway to involve in the enzymes that are presented in the cytoplasm, microsomes, and the peroxisomes. It is synthesis of the cholesterol like that. The most biological lipids begin from the two carbon acetate from the acetyl CoA. The initial step in the pathway of cholesterol biosynthesis are collectively called that mevalonate pathway, which are itself that and cumulative with the synthesis of the isoprenoid molecule and isopentenyl pyrophosphate will be formed, and finally will be a cholesterol will be the end product of cholesterol metabolism. So what is it? Show that the biosyn the liver approximately 10 percentages will be our synthesis, and the intestine also for 15 percentages in the amount of produces each day. The cholesterol biosynthesis for that in each day, the cholesterol biosynthesis, the enzymes or environments are presented in the first one is a cytoplasm, microorganism, peroxisome, a synthesis of cholesterol, and mostly the biological lipids. They begin from the two carbon, the combined together to form the acetate, acetyl CoA. The initial step of pathway of cholesterol biosynthesis are collectively called the mevalonate pathway. Just the cumulative will be the synthesis of the isoprenoid molecule, and finally to form this isopentenyl pyrophosphate, and finally will form this cholesterol. So this is the structure. We'll talk about for this and the summary about for the cholesterol biosynthesis. The cholesterol biosynthesis, first one is that to acetyl CoA and it acetyl CoA to form the HM-CoA. This process are known as HM-CoA formation. From the HM-CoA to convert it to form the mevalonate, this steps is called as for committed step. From the mevalonate to convert for isopentenyl pyrophosphate, this called as for the phosphorylation reaction. From the pharmacy pyrophosphate to the spinal, it is called as for this and condensation reaction, and finally will be a cholesterol will be a form that the reaction is called as cholesterol formation. From the cholesterol and then to enter into the three routes, one is to enter into the bile acid, and one more thing to synthesize for steroid hormone, and third one is to enter it for vitamin D. So what about that is mean the first form is HMCoA formation, second committed strip, third one is phosphorylation, and the fourth one is for condensation reaction, and the fifth one is for cholesterol formation. So there is a process. So the acetyl CoA are converted to form the 3-hydroxy-3-methyl gluteryl CoA, and agent CoA is are converted to the mevalonate second step. From the mevalonate is converted to the isopenide based molecule to form this isopentenyl pyrophosphate. From that isopentenyl pyrophosphate molecule to converted for the scalate. From the scalate to converted to form this and the final form is our cholesterol. So this is for that acetyl CoA. So the first reaction about this, so this is starting process for the cholesterol metabolism. The first reaction is our acetyl CoA to convert it to form this acetoacetyl CoA. And with the catalyzed by the enzyme, it is called as for thiolase enzyme. In this reaction is called as for acetoacetyl CoA plus acetyl CoA to form this HF CoA. By this enzyme, catalyzed enzyme is known as for HF CoA synthesis. This step it is called HF CoA formation. Again, there is the first reaction acetyl CoA to connect to form acetoacetyl CoA to convert the acetyl-CoA and with acetyl-CoA to combine it together to form this beta-hydroxy beta-methylglutamate-CoA that is called as the HM-CoA formation reaction. And the steps show. So this is for that, and initially for two molecular of acetyl-CoA, it converts to form acetyl-CoA, and for cystolic thiolase enzyme, from the acetyl-CoA to convert with further molecule for acetyl-CoA, it is catalyzed by HM-CoA synthase. It is called as for HM-CoA formation. So that is the reaction to acetyl-CoA to condense together to form the acetyl-CoA. From the acetyl-CoA to form the HM-CoA by the enzyme HM-CoA synthase enzyme. So that is with mevalonate for synthesis. The mevalonate, it is formed as, this is for mevalonate formation from this. It can HM-CoA is then converted to form this mevalonate by this HM-CoA reductase enzyme. It is bound to the endoplasmic reticulum. The NADH pH is act as a cofactor and the two molecules of NAD which are consumed during the conversion of HM-CoA to mevalonate. And the final steps are HMK to myelinate and catalyze the HM-CoA reductase. So this is for third step, HM-CoA reductase to HM-CoA to convert to form the myelinate, and with the help of the HM-CoA reductase enzyme, it is consumed by this 2 NADH, and to help by this acts for that 2 NADH, it acts as a cofactor in this reaction. And the third step is the conversion of mevalonate into activity for isoprenoid unit. This is for isoprenoid is containing for the molecule or important intermediate molecule of in the cholesterol biosynthesis. So this is for this the mevalonate to convert for and the mevalonate to convert to form that 5 pyrophosphomevalonate. This reaction to convert for the ATP are produces ATP catalyzed by this reaction, and this 5 pyrophosphate 5 pyrophosphomevalonate to convert to form this isopentenyl pyrophosphate and the release of carbon dioxide. In this reaction, for rollers for the and isopentenyl pyrophosphate will be formed. From the isopentenyl pyrophosphate, it is converted to form this the dimethyl pyrophosphate. There is the intermediate convert for isopentenyl pyrophosphate and the dimethylene pyrophosphate will be formed. So what happened thus? This is for the mevalonate and to form this here this isoprenyl group will be formed. The mevalonate to convert it to form this two step. One is for 5 pyrophosphate mevalonate and one more thing it is for the isopentenyl pyrophosphate. This is a two reaction. One first reaction will be to convert it for this 5 pyrophosphate to that isopentenyl pyrophosphate. The ADP will be produces and this reaction of carbon dioxide releases from this reaction. And one more thing, the isopentenyl pyrophosphate to convert from that dimethyl pyrophosphate that is made that intermediate product of cholesterol biosynthesis. And the steps for you know that is already polymerism of 5 carbon isoprenyl unit. Now to convert from that 30 carbon of linear structure of squalene. This is the most important step. The squalene will be formed. And how can to form this squalene? This means that dimethyl pyrophosphate to convert to form this sternyl pyrophosphate And from this, general pyrophosphate contains with for isopentenyl pyrophosphate. In this reaction, what about the pyrophosphate are releases to form this the final product in the paranacyl pyrophosphate will be formed. Each condensation involved and carboxylation is formed. Pyrophosphates are eliminated in this reaction. So, in this one, after the dimethyl pyrophosphate formation, from the dimethyl pyrophosphate to convert to form the gernyl pyrophosphate, the removal of isopentenyl pyrophosphate. The gernyl pyrophosphate and with it along with it attached for isopentenyl pyrophosphate to convert to form the sphingosine pyrophosphate. And this reaction is each condensation involved in this carboxylation form and pyrophosphates are eliminated in this reaction. And that again then two Paranacyl pyrophosphate now will be or form that squalene. From the squalene to convert to form that lanosterol. In this lanosterol, after the 16 reaction will be occur and finally will be a cholesterol will be formed. So that is mean that the two pyranols are here this and the parallel pyrophosphate means the two paranosyl pyrophosphate to convert to form the squalene. This squalene to convert to form the two three axial squalene. It means that accession will be occur from this accession to finally to form the lanosterol. So from the lanosterol and after the undergo for the 16th reaction and the 16th reaction and finally to form the cholesterol will be formed. And step 3, the sense of cholesterol from the lanosterol and 18 to 19 reaction will be occur and finally will be formed this cholesterol. That is the most important thing, the cholesterol will be formed. So this is for and cholesterol metabolism. What about here this? So I already told the first one is this is the structure. The first one is the two acetyl CoA to convert to form that acetyl CoA to form this acetyl CoA from by the catalyzing enzyme thiolase enzyme. Yeah. From that as acetyl CoA to convert it to form this beta hydroxy beta methyl CoA by this name is called by the catalyzed enzyme. We can the COSH are releases from this reaction by the catalyzed enzyme HMG CoA synthase. And for this here the and from that HMG CoA to convert to form the nevertheless in this reaction two NADPs are act as a growth factor and to NADPs are and relieved from this reaction from the regime CoA to convert to form the mevalinate and catalyze the enzyme HM-CoA reductase enzyme. In this step, and from this mevalinate, isoprene containing the molecule. It means that conversion of mevalinate activate the isoprene unit. So mevalinate will be formed and is attached for this isoprene now to activate it. And the isoprene containing for the molecule are important intermediate in the cholesterol biosynthesis. From this mevalinate to form that two reactions and one is for 5-pyrophosphomevalinate and one more reaction will be formed isopentenyl pyrophosphate. In this reaction, the first one is for ADP produces and second one is for 5-pyrophosphate to convert for isopentenyl pyrophosphate. In this reaction, the carbon dioxide are releases and the ADP will be or produces from this reaction. And isopentenyl pyrophosphate and converted for this the dimethyl pyrophosphate. This is for the reversible reaction and interconverted for isobutyl pyrophosphate and dimethyl pyrophosphate will be formed. And step 4 and final for the polymerization of 5 carbon atom isobutyl unit and to form this 30 carbon linear structure of this column will be formed. So that is known as from the dimethyl pyrophosphate to connect from this Genyl pyrophosphate in attached with for isobutyl pyrophosphate. From the genyl pyrophosphate to converted to form this and with along with for isobutyl pyrophosphate to form that paranaxyl pyrophosphate in this reaction and pyrite phosphates are eliminated from this reaction and to condensation involve the carboxylation form from the two farnesyl pyrophosphate to form and cannot to form the squalene. From the squalene, here to form the lanosterol and head-to-head condensation of two farnesyl pyrophosphate with the reduction by the NADPH and yield with the squalene sequence. From the squalene, it converted to form the lanosterol and from that lanosterol, we undergo for the 19th step will be there after that, the cholesterol will be formed, and this is the most important thing. And conversion of cholesterol to the cholesterol to involve in this 90 reaction and catalyzed by the enzyme in endoplasmic reticulum membrane. And addition, the modification yield the various steroid hormones and vitamin D will be activated by the cholesterol. So this is for and this is the metabolism for the cholesterol in the summary way and easy way to remember that. Now to move on to this regulation of cholesterol biosynthesis. It normally for that healthy adult to synthesize the cholesterol at the rate approximately for 1 gram per day and consume the approximately 0.3 gram per day and relatively the constant level for cholesterol in the blood 150 to 200 mg per day. It is maintained in the primarily by controlling the level of de novo synthesis. The level of cholesterol synthesis are regulated in the for by the dietary intake of cholesterol and cholesterol from the, both the diet and synthesis is utilized in the formation of membrane and in the synthesis of the steroid hormone and bile acid. The greatest proportion of cholesterol is used in the bile acid synthesis. Normally for this adult age the cholesterol approximately for 1 gram per day and consume approximately 0.3 gram per day and relatively the constant level of cholesterol in the blood 150 to 200 mg per dl and by the primarily are controlling the denosis of cholesterol biosynthesis. The cholesterol biosynthesis is a regulated part of the dietary intake of cholesterol. The cholesterol from the both the diet and the synthesis is utilized in the formation of membrane and in the synthesis of the steroid hormone and the bile acid. The greatest proportion of cholesterol is used in the cholesterol bile acid synthesis. And when the high blood cholesterol concentration is found in this diabetic mellitus, nephrotic syndrome, obstetric jaundice, and biliary cirrhosis and hypothyroidism. These are all the disorders. You can cholesterol biosynthesis will be are high. And low serum cholesterol levels are observed in this hyperthyroidism, malnutrition, malabsorption, and anemia. And physiologically, the lower levels are found in the children and worsening on the cholesterol lowering the drugs. And this is for the most important, how to the regulating the cholesterol biosynthesis, one name of the enzyme. Here is HM2A reductase enzyme. And what the reaction HM2A to convert to form that mevalonate and catalyzed by the enzyme HM2A reductase and 2NADH are utilized in this reaction. And the name of the regulating enzyme is HM2A reductase enzyme. In the model to over 100-fold as ranges will be are high. Again, that remember that HM2 reductase is one of the regulating enzyme. It is a regulating enzyme, not one of it is a regulating enzyme of cholesterol biosynthesis. And cholesterol and made in the liver is exported. Much of the cholesterol synthesis take place in the liver. The cholesterol exported in the two form One is for the bile salt. It is an amphibic cholesterol derivative that add the lipid digestion. And one was for cholesterol yeast. It is transported and secreted in the lipoprotein participate to other tissues that use the cholesterol or it is stored in the liver. So what about the cholesterol is made in the liver as exported one is for this cholesterol exported in the two form one is for bile salt amphibic in cholesterol derivatives that add the lipid digestion and the second one is for cholesterol yeast it is transported and secreted in the lipoprotein particle to other tissues that is use the cholesterol that is stored in the liver. The utilization of cholesterol and what are the function and utilized for the cholesterol is the cholesterol is transported in the plasma predominantly as cholesterol yeast is associated with the lipoproteins. And one more thing, the dietary cholesterol is transported from the small intestine to the liver with the help of that chylomicron and the cholesterol synthesis by the liver as well as many dietary cholesterol in the liver that exceed the hepatic needed to for transported in the serum with the serum between the LDL. The liver, it is synthesis the VLDL that are converted for the LDL through the action of endothelial cell. It is associated with for lipoprotein lipase and the cholesterol. It is mostly are found in the plasma membrane can be extracted by the HDL. So what are the uses for the cholesterol? One is for cholesterol is transferred in the plasma predominantly as the cholesterol ester. It is associated with the lipoprotein. And the second one is the dietary cholesterol is transported from the small intestine to the liver within the chylomicrons. And third one is for the cholesterol. It is synthesized by the liver as well as it may any dietary cholesterol in the liver that exceed the hepatic needed transfer the cholesterol from the within the help of the LDL. The liver synthesizes the VLDL and these are converted for the LDL through the action of endothelial cell and associated with lipoprotein lipase. The cholesterol is found in the plasma membrane can be extracted by the HDL. And next one is for this degradation of the cholesterol. The ring structure of the cholesterol cannot be metabolized easily, and now to convert it for the CO2 and H2 in the human, the intact the steroid ring is eliminated from the body by the conversion to the bile acid, which are exceeded in the fishes. And the second is the secretion of the cholesterol into the bile, which transported it to the intestine for the elimination. So, how to the degradation for the cholesterol, the conversion to the bile acid, which are excited in the fishes, and secretion of the cholesterol into the bile, which are transported it to the intestine for the elimination reactions. So, here that this is all again. Then summary about for this cholesterol biosynthesis. So, this is a specific learning objective from this topic and the cholesterol metabolism. And first, we talk about the structure, and the second one is talk about for the metabolism and the biosynthesis of cholesterol. And third one is for function and sources for cholesterol biosynthesis and variation of the serum. It means that what is the normal level of cholesterol and the utilization and how can the cholesterol are degraded and final one is how to regulate and for this cholesterol. So this is for the introduction about for the cholesterol. We know that the cholesterol in the animal tissues, man is in the animal tissues, and cholesterol it is present in the tissue and in the plasma are either are present for the cholesterol in nature, free cholesterol in nature, or is present for a cholesterol ester, the long chain fatty acid. Combined with the long chain fatty acid, it is called as for the cholesterol, cholesterol ester. And one more thing, and remove from the, it is why most important thing why that cholesterol HDL is a good cholesterol and VLDL bad cholesterol. That is why it is removed from the tissue by plasma high density lipoprotein and transport to the liver, and where it is are eliminated from the body either the unchanged or after the conversion of bile acid in the process is known as reverse cholesterol transport. This is for cholesterol structure having for that four aromatic ring A, B, C, D and 27 carbon atoms are presented. It is presented for this for C5 and C6 and one double bundles are presented, one side chain and one more thing for this hydroxyl groups are presented in this C3. This is the sources and the maximum I get it from the diet and the de novo synthesis and itomasis for cholesterol ester approximately for 10% for each of the total synthesis like human liver and the intestine account for approximately for 10 percentages synthesis for the cholesterol in the human and usually all the tissues containing the nucleated are capable of cholesterol synthesis, which occur in endoplasmic reticulum and cytosol. And the cholesterol is the most abundant steroid in the human and performing the number of essential functions. The first one is the major constitution of the plasma membrane, and it is a precursor of bile salt. And third one is a precursor of steroid hormone, like for radical hormone, sex hormone, and pleasure hormone. It is also the precursor of vitamin D, and it also to record for the nerve transmission. And what are the steroid hormone production from this? It is a synthesis for cortex, adrenal gland. The two and classes of hormones are synthesis. One is for mineralocorticoid and the glucocorticoid, and sex hormone like for progesterone and androsterone will be produces from this and the biosynthesis of cholesterol. So this is the most important thing. What about that is slightly less than half of the cholesterol in the body derived from biosynthesis by de novo synthesis of cholesterol. And biosynthesis in the liver account approximately for 10 percentages, but intestine approximately 15 percentages. Clear? And the synthesis of cholesterol, like that, most biological lipid and begins from the two carbon acetate group of acetyl CoA. And this is the initial step or pathway for cholesterol biosynthesis. And collectively called as the melanin will be formed. From the melanin to be converted to form this isoprenoid molecule. And from the isopentenyl pyrophosphate and finally will be formed cholesterol. So, this is the summary about for the cholesterol. First one is for HM-CoA formation, and the second one is for committed strip, and third one is for phosphorylation reaction, and the fourth one is for condensation reaction, and the final one is for cholesterol formation. Okay, so and one is a two acetyl CoA to convert to it is converted to form those acetyl CoA, and from the acetyl CoA to convert from this HMG. This reaction it is called as for HMG HMG-CoA formation. This is called as from the HMG-CoA to convert it for this mevalonate. This is for the committed state. From that mevalonate to convert it for that isopentenyl pyrophosphate. This is for the phosphorylation reaction. From that isopentenyl pyrophosphate to convert from the balanced pyrophosphate. This is for the condensation reaction from the balanced pyrophosphate converted from the squalene, that is mean that condensation reaction from the squalene to form the cholesterol. This is mean that cholesterol formation. From the cholesterol, one is from the bile acid, second one from the steroid hormone, and the third one to form this vitamin D. And most important thing is the rate limiting enzyme for this pathway, HM-CoA reductase. And this reaction from this acetyl-CoA to convert from the HM-CoA. It's a malonyl, sorry, that's a HM-CoA to convert to form this malonyl and catalyzed by the enzyme name as for HM-CoA reductase. So we already told this is for the first one is secondary formation, second is the malonate formation, and third one is for isopentenyl pyruvate phosphate formation, squalene formation, and cholesterol formation. So that is the first reaction. Initially for the two acetate from acetyl CoA from the acetyl converted from the HM CoA by the enzyme HM CoA synthase, and it second step from the HM-CoA and release of NADP and utilize a cofactor like for 2 NADPH to convert to form this mevalonate and to by the enzyme name is for this HM-CoA reductase. This is the one of the rate limiting enzyme. This is that biosynthesis of cholesterol biosynthesis, rate limiting enzyme of cholesterol biosynthesis. And third reaction, the mevalonate will be formed. And one more important component that will be the isopentenyl, that is for isopentenyl, isoprenyl ring will be formed. This isoprenyl containing the molecule are important for the intermediate in the cholesterol biosynthesis. In this mevalinate converted for this 5-pyrophosphate mevalinate from the 5-pyrophosphate mevalinate converted for the isopentenyl pyrophosphate. This is the two step, and this steps are two carbon dioxide are releases there, and one more steps are two ADP are releases from this reaction. So the isopentenyl pyrophosphate and the intermediate compound of dimethyl pyrophosphate will be formed from this in this reaction. Dimethyl pyrophosphate along with for isopentenyl pyrophosphate to form this genuine pyrophosphate and release of pyrophosphates are releases from the genuine pyrophosphate to form and along with together isopentenyl pyrophosphate and to finally to form the farnesyl pyrophosphate in this reaction, and pyrophosphates are eliminated in this reaction. From that farnesyl pyrophosphate and one more farnesyl pyrophosphate between the two farnesyl pyrophosphate to convert for the squalene, and this is for reduction by NADPH and finally to form that held by the squalene with and intermediate will be formed this lanosterol. This lanosterol undergo for this 19-step reaction to connect to form this cholesterol. In this conversion of cholesterol, involved the 90 reaction and enzyme are present for endoplasmic reticulomembrane. Additional modification held the various to form steroid hormone and vitamin D. And this is for that regulation of the cholesterol biosynthesis. You know that, and normally for this, approximately for the rate of the approximately 1 gram per day and consumed approximately and 0.3 gram per day, and relatively the constant level for the cholesterol in the blood 150-200 mg per day. Now we are changes and 230 and primarily and controlling by the denominations of cholesterol, and the level of cholesterol synthesis are regulated in the part of dietary intake of cholesterol. Again detail the cholesterol only are controlled by under the diet and drugs. Clear. The cholesterol from both diet and synthesis are utilized in the formation of membrane and in the synthesis of steroid hormone and bile acid. This greatest proportion of cholesterol is used in the bile acid synthesis. And what are the conditions of high level for cholesterol? One is the diabetic mellitus, nephrotic, obstructive jaundice, and familial hypercholesterolemia and biliary cirrhosis and hypothyroidism. This is that cholesterol levels are decreases and hypothyroidism, malnutrition, malabsorption, and that anemia cases and physiologically lower levels are found in the children in person of cholesterol lower in the tracks like. So that is a strict, we already told HM-CoA reductase, it acts here. HM-CoA acts for that rate limiting step of cholesterol biosynthesis. This is the cholesterol made in the liver, the exported, and the two way one is bile acid formation, and second one is for cholesterol esters. And the utilization of cholesterol, you can already know that one is to transfer to the plasma predominantly as the cholesterol ester associated with the lipoprotein. And the dietary cholesterol is the transported from the small intestine to the liver with the chylomicra. And one more thing, the cholesterol synthesized in the liver by the liver, as well as the dietary cholesterol in the liver that exceed the hepatic need is transported from the within the LDL level. The liver synthesis of VLDL and this was converted for LDL and in the endothelin cell associated lipoprotein lipase enzyme. The cholesterol found in this plasma membrane can be extracted by the HDL. And one more thing, the ring structure of cholesterol cannot be metabolized CO2 and H2 in the human. The intact steroid rings are eliminated from the body. One is the conversion of bile acid, and the second one is secretion of cholesterol into the bile. So, this is all for this and some report for the cholesterol biosynthesis regulation and it, cholesterol biosynthesis regulation extracted and what are the other structures, all are discussed about. These are all about it for this and summary about for the classes. You can go and read and open your book and if you have any doubt, please come and talk with me. Thank you.